Okay, thank you so much for the opportunity to be here today. I've been attending the session and this really, really exciting data on clinical trials and clinical development. Today we'll provide you an overview of clinical development in gene therapy while presenting the experience of applied stem cell therapeutics in gene therapy, gene editing, and cell therapy. I will describe the key components of our gene therapy for hemophilia A. I will describe the elements like viral vectors, transgene, and promoter, preclinical data. I will try to do that in a way that you're going to, for those not familiar with this space, being able to understand how are we dealing with this type of drug agents. I will also explain the advantages of using in vivo gene editing using CRISPR-Cas9 for hemophilia A, and you will be able to learn about unique operational aspects of gene therapies. Okay, let's go for it for this journey from the lab to the hepatocyte. And let me start with hemophilia A. This is a rare genetic disease affecting the synthesis of factor VIII by the hepatocytes in the liver. Patients lack this clotting factor and can suffer serious bleeding episodes and require therapy for life. In the US, there are about 40,000 patients, 400,000 worldwide. Now, to describe the progress achieved in gene therapy for hemophilia A in the past 20 years, I am using as analogy the Apollo 11 historic journey that I'm sure most of you are very familiar with to land a man in the moon. Remember that it required a rocket with a propulsion capsule that you can see in the slide to the right, upper right, and with a lunar module. For gene therapy, we also need a rocket. This is the viral vector, which carries a payload consisting of a promoter, the propulsion capsule, and a transgene, which is the lunar module. In traditional gene therapy, and this is what you see at the bottom right, the promoter and the transgene are orbiting the genome, meaning they are not integrated into the genome. But in gene editing, there is an actual landing or integration in the genome using the CRISPR-Cas9 technology. Okay, and here, by the way, this slide is the actual slide of the lunar module after it was left behind in the surface of the moon. I know the audience has nowadays a significant knowledge about viruses, especially the one at the bottom right. For gene therapy, we need a viral vector as a rocket to reach the target cells. In hemophilia, is the adeno-associated virus, shorthand AAV, also a spiky virus, as you can see, but completely harmful to humans. It has a navigation system that preferentially reaches the liver. It enters the nucleus of the hepatocyte without integrating into the host genome, unless specifically directed to do so. That's why it's called this situation episomal, and it's called an episomal single-stranded DNA, which is lost through cell division. So that's one of the reasons why this type of approach, gene therapy, in an episomal format, is used for adults aged 18 years and older. We're going to see in a moment why. There is some immunogenicity, and we're all hearing about antibodies nowadays for the virus. But here it depends on the type of serotype that you use to see what type of immune response you see to the virus. I discuss here the development milestones of ASC 618, which was originally developed by scientists and clinicians at Emory University and exclusively licensed to ASC Therapeutics. Like with the propulsion capsule and the lunar module in Apollo 11, here we also have the promoter and the transgene. Once the viral vector AAV8 enters the hepatocyte, it releases a liver-specific promoter and an engineered transgene for factor VIII. The promoter activates the expression by the transgene of the factor VIII. And this protein structure is shown here to the right. You can see that, obviously, I can tell you that this conformation is different from the standard protein because the B domain has been deleted. You can say A1, A2, A3, C1, C2, but not B. The reason, and it was already done 20 years ago when hemophilia A and hemophilia B, by the way, started being developed, they realized that in order to get both elements, the promoter and the transgene, into the AAV viral vector, it had to have a maximum length close to 5 kb. And that was impossible with the B domain. So that's why it was deleted. And our construct also has this B domain deleted, but it has certain characteristics that we're going to see in a moment that make it more efficient in terms of synthesis and secretion. And that's because the group of Emory University discovered certain ancient sequences in the porcine factor VIII that were shown to increase, again, synthesis and secretion. And here is some data showing that effectively the ASC 618 is more efficiently synthesized and secreted compared with native human factor VIII. Is again a bioengineered construct. It has what is called a liver codon optimized transgene because it includes these 9% of porcine sequences, and the promoter has the minimal length possible to ensure that we get this efficient construct. In this line graph, we show that the promoter and the transgene of ASC 618, the upper line, when injected intravenously in mice, are more efficiently biosynthesized and secreted than standard factor VIII constructs in the lower line. And these are the ones that we're going to see in the next few years approved, by the way, like the gene therapy from BioMarin and Sangamo. So, why am I sharing with you preclinical data? We're here to discuss clinical trials, and I'm the Chief Medical Officer. I'm responsible for the clinical development program. I think is, based on my experience, is extremely important to ensure that I am really embedded into what we're seeing in different animal models. When we had our conversation, our pre-IND meeting with the FDA last year, they told us, which is a standard, that we needed now to move from just one animal species, the wild type mice, into also humanized mice and non-human primates. All three are considered different species. The humanized mice incorporates human liver cells and is considered, again, a different type of species. What we saw in our results is that first, the factor VIII protein levels are significantly elevated in a dose-dependent manner. That's important to see that effectively we're seeing this increase over time, depending on the dose. That indeed there are some factor VIII protein inhibitors that were well known in the industry because up to 25% of patients that receive any kind of replacement therapy with a factor VIII protein develop these inhibitors, and we also see them here. And they also indicate that there is a successful factor VIII protein production following ASC 618 treatment. And extremely important, of course, is especially non-human primates to the fact that we didn't observe adverse events like changes in body weight, body temperature, clinical signs, liver enzymes, this is extraordinarily important, and other hematological parameters. So in summary, our gene therapy program had an extensive discovery program to create the viral vector construct with a patented and published promoter and transgene sequence. You see here the sequence at the bottom left, and again, is exactly the length that this is required to be able to incorporate that into the viral vector, our rocket. And we have many publications, among them these that I have here, showing indeed how this type of approach works in animal models. We had an interaction with the FDA, we also got an orphan drug designation granted by the FDA. We expect to initiate the phase 1 clinical trials in the first quarter of 2021, and I'm, as you can imagine, very involved in ensuring that we get our IND package ready, incorporating all these preclinical data, the CMC manufacturing details, and all the planning, study design for our clinical trial, phase 1 clinical trial. Now, let's now take a different take in terms of moving now from just inserting the gene into the hepatocyte and being able to produce the factor VIII in these patients with hemophilia A without integrating the gene into the genome. Now we're going to take the approach of inserting the factor VIII transgene into the genome. And this is the so-called gene editing approach. For our program is called ASC 518. It's a separate clinical program that uses the same bioengineered transgene for factor VIII, but this time integrating it into the genome. It's like landing the lunar module to send radio signals indefinitely from the surface of the moon. Our research team has developed a CRISPR-Cas9 based gene editing technology. It was published in Nature Scientific Reports, which is, as you can see here, and it uses 2 steps. First, we inject the viral vector AAV8 containing the CRISPR-Cas9 enzyme with a guide RNA that identifies a unique DNA sequence along the albumin locus, especially important in the hepatocyte. It makes a precise cut like genetic scissors. And next we inject another viral vector, AAV8, similar to the one that we used for gene therapy, but in this case containing only our proprietary transgene without the promoter. So we have demonstrated in this publication that the promoter of the albumin locus activates the transgene to synthesize and secrete factor VIII in animal models. So why is it important to use this other approach, these genetic scissors, and to insert the gene in a certain place instead of just having it orbiting the genome of the hepatocyte? The main reason is because of the lifetime effects. So it makes the synthesis and expression of factor VIII independent of hepatocyte turnover. And this is especially high in the pediatric population. So in contrast to gene therapy, gene editing allows the integration of the transgene in the genome and is always available. Additionally, we are seeing in our case that this construct is highly potent and efficacious. It takes advantage of the highly active albumin promoter in hepatocytes, and it drives the high levels of factor VIII. And there is a site-specific integration in the genome so that it doesn't have off-target effects, which would make it not safe. So now, to ensure that the entire discovery, preclinical and clinical development program truly translate into a successful treatment for patients, gene therapy and gene editing programs have unique challenges and complexity, which explain why these therapies can reach over $1 million for a single infusion. Obviously, this is once and done. So the moment that the patient responds to your injection, you follow up the patient, but there is no more treatment needed. This is a cure in principle. And I think that gene therapy requires unique expertise and coordination for manufacturing and clinical studies. And that's why I'm using as an analogy the Houston's mission control for Apollo 11. Now, for those of us who have been in clinical development for some time, we have experienced a correlation of the molecular size of the therapeutic agents with their manufacturing and characterization complexity. I started my clinical development career with a biologic, AbbVie's Humira, about 20 years ago, and we felt at the cutting edge. I sensed like being a pilot in a fighter jet. From the time I joined Avexis clinical team about 5 years ago, developing the gene therapy Zolgensma, also working with Unicure and Synvivo in gene therapy, to my current role with ASC Therapeutics, I now feel like an astronaut in the space station and just being part of an entire team of people that I need to be very, very aware how everything works in this space station. So, as a representative example of the complexity to produce a viral vector, every company needs to develop sophisticated processes to facilitate manufacturing. And I'm really very aware of everything and how all the different steps are working here. For ASC 618, we introduced 3 DNA sequences or plasmids, which are the transgene, the promoter, and structural viral genes, that all together form a kind of assembly line into a producer cell, well known in the industry, called HEK293. And this cell starts producing viruses. There are 2 ways to look into these viruses. Either and we call what we get produced by these bioreactors a soup. The soup includes a supernatant and also the pellet. We find different levels of AAV in both of them. And again, these are AAVs. These are viral vectors that already incorporate the transgene that we use. We need to understand, especially me as Chief Medical Officer and responsible for clinical development, I need to understand what is the potency, the yield, and of this kind of manufacturing process. I need to use that to ensure that I understand what is going to happen throughout the clinical development period. I also must understand the way, the analytical methods to measure viral titers, viral genomes in the final drug product are going to inform the methods I use for my gene therapy clinical programs. So I'm trying to give you a sense that what we are doing in gene therapy and gene editing is really unique in contrast to the way we have been developing drugs in the past. And then finally, I want to show you an overview of the critical elements of our hemophilia gene therapy clinical program. We are first selecting a severe population of patients who will benefit the most from a single infusion that will actually cure their disease. A single infusion that will change their lives. No more factor VIII replacement therapy or treatments for bleeding episodes. Besides the standard phased clinical program, phase 1, 2, 3, we also, the FDA requires us to follow the study patients for a minimum of 5 years. There are a number of selected endpoints that I've included here, and in terms of safety, it's very important to assess the viral vector infectivity so that we can exclude off target in other organs, although they happen at a lower level. We want to understand the existence of total and neutralizing antibodies to AAV8 in our case, and the so-called factor VIII inhibitor level. So antibodies that block the actual factor VIII protein that we know happens with any type of therapy. So we need to look into all these elements when evaluating the effect of our therapy in these patients. And finally, we definitely need to look into factor VIII activity and also changes in bleeding episodes, recurrent treatment, and how it's impacting replacement therapy. So before finalizing, I just want to make a few comments on some of the conversation that we had before, especially the panel, when we were talking about how to look in this situation with the virus, in terms of remote monitoring, and we're looking to that. We have right now 3 bits for CROs that are going to help us with the phase 1 clinical trials, and we're asking them specifically to tell us how are they going to do this remote monitoring. Obviously, the patients will need to be in the clinic for the first infusion and staying in the clinic for the first maybe up to 24 hours. But the next visits potentially could be done with traveling nurses or with teleconferences or video conferences with the physicians. We are evaluating that, and I think that's something definitely important. Thank you so much, and happy to take your questions.